Hi everyone. In today's video, I'm going to go through the process of DNA transcription in eukaryotic cells. So firstly, what is DNA transcription? Well, it's one of the key processes within a cell and essentially is the process of converting DNA into RNA. And RNA is a key precursor in the formation of proteins, which we all know are the main molecules that define the structure and function of a cell. So how does DNA transcription start? So firstly, I would just like to draw a little bit of a comparison between prokaryotic and eukaryotic cells. So if we have a DNA strand here, and say this is a eukaryotic cell, and a DNA strand here, and say this is a prokaryotic cell. In a eukaryotic cell, the enzyme called RNA polymerase II is what initiates the process of DNA transcription. On the other hand, in prokaryotic cells, this enzyme is simply called RNA polymerase. The structures of these two enzymes is quite different as well. Firstly, the RNA polymerase II enzyme in eukaryotic cells, in order to be activated, must be phosphorylated. So if we have the enzyme here, for example, which is not attached to any DNA strand yet, it must have a phosphate group attached to it in order to be activated. On the other hand, RNA polymerase in prokaryotic cells does not have to be phosphorylated. The processes in which these transcription processes start is quite different as well. As we all know, in a DNA sequence, there are many different important parts. Firstly, there's the promoter sequence, which is seen in both prokaryotic and eukaryotic cells. Then we have the 5' untranslated region, which acts as a buffer and can also help in the stability of the mRNA molecule. Then we have the coding region, which contains both introns and exons in eukaryotic cells, but only contains exons in prokaryotic cells. So write it in red here. So only contains exons. Now, after this coding region, we have the 3' UTR region. And this has a similar role to the 5' UTR region and essentially allows for stability of this DNA chain and also allows for stability when the RNA chain is attaching to the ribosome in order to allow for the process of translation to occur. And after this, finally, we have the termination sequence, which follows the stop codon, or stop sequence. The termination sequence is a sequence of nucleotides that essentially tells the RNA polymerase molecule to stop its transcription process. And this is present in both prokaryotic and eukaryotic cells. Now, the process by which RNA transcription, or DNA transcription, starts in prokaryotic cells is different to eukaryotic cells as well. If we take prokaryotic cells, for example, and redraw our diagram here, firstly, prokaryotic cells actually have what we call a sigma factor joined to their RNA polymerase enzyme. And this sigma factor is what allows for, firstly, the RNA polymerase molecule to join to the coding strand of the DNA. Sorry, this should be the non-coding strand. And once it encounters the promoter sequence, It allows for the RNA polymerase molecule enzyme to attach more tightly to the DNA chain. And after this process occurs, it essentially just removes itself from the RNA polymerase enzyme and therefore allows for the process of transcription to occur. In eukaryotic cells, on the other hand, the process is a little bit different. If we take, for example, the strand that we drew before and redraw it here, there's no sigma factor present in eukaryotic cells. Instead, the RNA polymerase II enzyme is phosphorylated in order to activate it. So we have a phosphate group added, and there are special molecules called binding proteins, or they're sometimes even referred to as transcription proteins, and these essentially allow for the RNA polymerase II enzyme to bind more closely to the non-coding strand of DNA when they encounter the promoter sequence. Now you may have realized that I mentioned that the strand that the RNA polymerase enzyme attaches to is called the non-coding strand. This strand is essentially the strand that the RNA polymerase II enzyme binds to in order for the transcription process to occur. And there are two types of strands in all cells in DNA, the non-coding strand and the coding strand. The coding strand is the sequence of DNA that the RNA chain is corresponding to, whilst the non-coding strand is the complementary strand. So say, for example, we have a particular random DNA sequence which is A, T, G, C, G, T, G, A, C. The coding strand, which is what is the RNA sequence, will be the exact same, except the T's will be replaced with U's, because we all know that RNA has uracil instead of thymine. The non-coding strand, on the other hand, will have the complementary DNA sequence. So it's essentially the other side of the DNA that will have the complementary of the coding strand. So in this case, this will have T, A, C, G, A, C, T, G, because this is complementary to the opposite nucleotide base. Okay, so that's it in terms of the structure and how the actual process of transcription is started. Let's get into how this process actually occurs. So I'll only be going through this in terms of eukaryotic cells. So firstly, we have the RNA polymerase II enzyme, which is phosphorylated in order to be activated, and it reaches the promoter sequence. This promoter sequence is essentially what allows for the RNA polymerase enzyme to bind more closely to the DNA sequence, and also allows for the transcription process to start, and it also allows for the controlling of the speed of transcription. So it can allow for transcription to happen fast or slow. So I mentioned earlier that after the promoter sequence, there's also the 5' UTR sequence. The 5' UTR sequence is not really that helpful in terms of coding. However, I did mention earlier that it allows for stability. So what do I mean by that? So as we all know, that the process of transcription and translation requires for a particular DNA sequence to move around a lot in a cell, and therefore when it binds to specific organelles such as the ribosomes, it must be able to withstand a lot of changes in conformation and movement. So essentially this sequence basically allows for the RNA sequence to withstand all of that pressure. And it also allows for the RNA sequence to properly bind to the ribosome in order for the process of translation, which I'll explain later, occur. After this, we have the coding sequence, and this is the most important sequence in the DNA strand, as it will be what essentially will code for the protein. The coding sequence, as I mentioned earlier, contains introns and exons. Introns are, for this video's sake, simply nonsense strands. They are present in the coding strand between the exons, but they don't really code for anything in terms of a protein. What the coding strand actually looks like is something like this. So if we take, for example, the rainbow sections as the exons, they will be the parts of the DNA sequence that will code for a specific protein, say for example hemoglobin. On the other hand, the gray sections of the DNA chain are the introns. And scientists don't actually know why these introns exist. It's believed that they allow for stability. However, their function is not fully known. So they don't actually code for anything in particular in terms of the protein. So after the RNA polymerase enzyme is able to run through the sequence from a 5' to 3' direction, it's able to code for both the introns and the exons. After the coding sequence has been surpassed, it enters the 3' UTR sequence. The 3' UTR sequence is similar to the 5' UTR sequence, as I mentioned earlier, and it essentially just allows for stability once again. After that, we have the termination sequence. The termination sequence is essentially just the part of the DNA strand that tells the RNA polymerase enzyme to stop transcription and dephosphorylates it. And as we all know, dephosphorylation is what results in the inactivation of an enzyme. So the RNA polymerase II enzyme essentially just breaks apart and is waiting, therefore, for its next phosphorylation in order to allow for transcription to occur once again. So you may be wondering, great, okay, so that's the process of transcription as a whole. But what happens to the RNA chain after that? So I did mention earlier that there's a sequence of introns and exons, and let's say that this is the DNA chain, or the coding sequence of the DNA chain. The RNA sequence will have a similar structure in that it will have introns, which I'll denote in red, which will be at the same positions as the exons for the DNA sequence. So exons on RNA equals exons on DNA. And in this color, I'll denote the introns, which will also be in the same position as what they were in the DNA chain. So this will just allow us to draw the process more easily. So what happens after that? Essentially, this is not the final or the preferred form that the RNA chain is in order to code for a protein. So we have to make some modifications. And there are three main modifications that take place. One is called 5' capping. The other is called polyadenylation. And the third is called splicing. So let's first start off with 5' capping. 5' capping, as the name suggests, occurs on the 5' end of the molecule. So, say for example, the only sort of starting codon sequence that occurs in all RNA sequences is AUG, and this codes for the amino acid methionine. AUG is the start codon and essentially just starts the process of transcription to occur. However, having it open in this space doesn't make the RNA molecule that stable, so we need to add a methylated guanine group. And this is what 5' capping is. By adding another guanine group with a methyl group on it, we essentially allow for stability on the 5' end of the molecule. There's a similar idea on the 3' end, and that's what occurs in polyadenylation. Polyadenylation is a similar process in that it allows for stability on the opposite side. And now there are 3 different types of stop codon sequences that can occur. These are UAA, UAG, and UGA. After any of these 3 sequences, we can have a long string of adenine groups, and this can be in excess of 125 nucleotide bases in a long chain, what we call a polyadenine trail or tail, or poly-A tail. And this also allows for stability of our RNA molecule because essentially as it's capped on both sides now, it's sort of like being protected on both sides and it's able to be transported in a better manner. So now, after this, the third and final stage is called splicing. Splicing is a very important stage because it essentially gets rid of all of the unnecessary genetic material that we have. And as I mentioned earlier, for the purposes of this video, introns are essentially nonsense strands. So we have special molecules that are called spliceosomes. And they are essentially RNA molecules, and they allow for the cutting of the spaces between introns and exons. So they cut between the introns and exons in order to separate them and eventually allow for the RNA molecule to be just made up of exons, because they also allow for the exons to be joined to one another. So now is what we call the formation of a mature mRNA molecule. This mature mRNA molecule is what will eventually code for the protein. And in terms of the introns that we took away, these introns essentially go through a complicated process in which they form a loop because they are cut away from the intron structure, and the formation of this loop is very important because it allows for the intron to not interfere in any other process that occurs in the cell, and eventually they're disintegrated. So that's the process of transcription in a nutshell. We first start off with the appropriate DNA strand. Then we have an RNA polymerase molecule that runs across the DNA from the promoter sequence to the termination sequence, and that allows for the RNA molecule to be formed. And after some post-transcriptional changes, because they happen after transcription, or modifications, we have the formation of a mature mRNA molecule. And this mature mRNA molecule can pass through nuclear pores in the nuclear membrane that's present on the outside of the nucleus and pass out of these pores in order to reach ribosomes and undergo the process of translation. And you can check out my other video for the process of translation. Thanks for watching.